Alright, so yeah, so we've got these three different types of genes, oncogenes, tumor suppression, and DNA repair genes that are probably involved, at least in some small way, with cancer. Because a mutation to any of these genes will cause loss of mitotic control. So how do we kind of get to this point? So again, more research recently discovered probable epigenetic cause of cancer. If you end up with DNA methylation, so a methyl group attached to a tumor suppression gene, this will cause the gene to switch off, which will reduce the monitoring of uncontrolled mitosis. We might also have some kind of epigenetic change to repair gene. So perhaps instead of methylation, we've got some histone winding. So that gene is bound up to a histone protein or a histone complex, and we don't end up with the gene being expressed. Or perhaps even something's gone on and we've got some exon splicing. So maybe we talked earlier about the factors that increase our risk of mutations. So let's say we've got a viral infection, a hepatitis or HIV or whatever. We've got a viral infection, and that viral infection is producing an miRNA molecule, microRNA, remember those? And one of the things we said about microRNA is that they could be involved in this splicing process. So let's say that this viral infection is producing a microRNA protein, or a microRNA segment, sorry, not protein at all, a microRNA segment that is attaching to the exon for your DNA repair gene and causing it to be spliced out. So any one of these three steps that we've got up on the board could have an effect. What we're finding more and more is that attempts to treat cancers have been utilizing epigenetic control to up-regulate the body's own response to tumors. I did actually see this come up, I think, this idea come up in a few different examples in your SHE tasks. We won't go into the discussion in detail now, but I do just want to bring it up. Oh, good. That's very helpful. So we're not going to do the discussion now because we're obviously needing to accelerate a bit. But what they've done in this process, is they've extracted a patient's T cells, or in this case it's mice. But they've extracted T cells, which are a part of our immune response. And the body treats cancer, if it can detect it, treats it like a disease. So they remove the T cells, and then they activate the T cells. They modify the epigenome, not the genome, because if they modify the genome, that's called transgenesis or genetic engineering, gene editing. They're not editing the genes, they're modifying existing genes. They're turning certain genes on or repressing other genes. And they're doing that so that the T cell is able to detect the cancer. So this is, and you see how recent this is, January 31, 2018. So this is incredibly new medicine that they're talking about. Most of the time, by the time it gets to the media, just as an aside here really, by the time it gets to the media, we're still probably 10 or 15 years off of this actually being used in a clinical stage. It might be at clinical trials, but if you were to develop cancer, you're probably not going to be able to be treated with this for another 10 years or maybe even 15 years. And another aside, it often costs somewhere between a few $100 million to $1 billion to develop something like this to the full rollout stage. And that's including things like, how do you get it to an industrial scale where you're producing enough of this. We've got 1.5 worksheets, so key terms, page 69, questions 1 to 12. I hope everybody's keeping track of all of those. Did you have a question? Or? No? All right. Key concepts there, they're already up on the, yes?
With what you were saying about the T cells, I did that for my SHE test last year.
Oh, okay. Was that the, oh yeah, CAR T cells, yeah. That was a year 12 we did that as well. I'm not sure. I don't remember exactly what that article said. I remember the bits and pieces. I think CAR T was, you were up-regulating something, weren't you?
Yeah, it wasn't taking out the actual T cells, but it was making a T cell.
And then inserting it in. Okay. I might have remembered wrong. That might have been what they were talking about there. But so it'd be a similar sort of principle at the very least. All right. Oh, we've got to be close. We're not. Okay, gel electrophoresis and DNA sequencing. So in this, what we're going to look at is DNA sequencing process. So how it. Let's have a quick break actually.
Thank you.
Natalia, would you mind just pausing the video? It's only been running for a few minutes.
I got a quick question.
Yes.
So we talked about yesterday.
Yes.